Aloha everyone, my name is Sara Wood and I am graduating from the Masters of Science Marine Science program at HPU. I am presenting my research project I did as an intern with a local nonprofit, Maunalua. This is Finding the Fins, a habitat assessment of Maunalua Bay. For this journey, I'd like you all to join me on a tour of Maunalua Bay, located on the south shore of Oahu. For my study, I completed snorkel surveys at the 5 locations you see here in yellow, all part of the greater Maunalua Bay, which extends approximately 5 miles across and is located between 2 iconic landmarks, Diamond Head and Cocoa Head. Because of its topography, Maunalua Bay is the catchment for numerous watersheds, making it vulnerable to detrimental land-based activity. My study serves to better understand how the ecosystem has been impacted by the issue. The overall goal for conservation is to restore healthy coral reef ecosystems in Maunalua Bay. Coral reefs are incredibly valuable. Statewide, they are worth over $33 billion to our local economy. They provide habitat, offer protection from storms, provide food for our community, and attract tourists to stimulate our economy. However, our reefs are under threat. They face anthropogenic challenges like pollution, global warming, ocean temperatures that are rising and can lead to coral bleaching, increased human infrastructure and development, and overfishing, which removes key organisms from the food chain. The loss of our coral reefs will have cascading effects, resulting in loss of biomass and resiliency. For my project specifically, I really wanted to focus on strengthening the existing monitoring efforts and community engagement that Malama Maunalua has been doing. My primary questions for this project are to determine where the majority of fish are and if their presence has any correlation to physical factors like reef infrastructure or site location. This information can then be used to inform policy decisions, especially since Maunalua Bay has been proposed as a site for a fisheries management area where fishing may be restricted in the future. All of our surveys were completed in the nearshore habitat in areas accessible by swimming. To complete these surveys, 5 snorkelers swam in a straight line transect for 5 minutes. Transects were run parallel to the shoreline and as close to the reef break as possible. The first snorkelers were the fish counters and tallied the number of fish encountered on the survey plate you see here. Rather than recording species, we grouped fish by families such as wrasse, damselfish, and butterfly fish. The middle snorkeler swims behind the first 2 and takes photos of the bottom habitat every 10 feet. You can see some examples of what the bottom habitat looks like at a few of our sites here. These photos will be uploaded to an online database called CoralNet for further analysis. Lastly, 2 snorkelers follow at the end to record coral abundance and health. They make note of every live coral head they see and match the color of it against the ko'a card, which was developed by researchers at the Hawaii Institute of Marine Biology to monitor coral bleaching and degradation. When sea temperatures are elevated, corals will expel their symbiotic algae, causing them to bleach and turn white. This card helps to document these stages of bleaching and monitor recovery. To best assess the habitat of Maunalua Bay, the data collected during the summer has been combined with previous efforts from spring of 2022 for a total of 33 transect sites across 11 sampling locations seen here. The majority of fish recorded were wrasse, which make up almost half of the fish seen in Maunalua Bay. The next most abundant were surgeonfish, representing about 1/5 of total fish, and the remainder of the groups were more similar in abundance, with none exceeding 7% of total abundance. As mentioned before, one of my primary objectives was to get a better understanding of where the highest concentrations of fish are in Maunalua Bay. By far, Black Point West, which is located to the northeast of Diamond Head Crater, had the highest fish abundance. This area also had the most pristine, intact corals. So it's likely that the infrastructure of coral reef ecosystem in this region contributed to the higher fish abundance. The lowest fish abundance was seen at Wailupe, which is a site with a strong reef break which may inhibit reef growth. I completed a Pearson correlation test between variables and found a positive relationship between the fish abundance and species diversity. In an ecosystem-based management, diversity is a really important consideration because it has been linked to ecosystem functioning and productivity. Therefore, this information and continued surveys could be used when considering management options. To assess benthic or bottom habitat, we entered our photos into an online database called CoralNet. This system is used by researchers worldwide and across the state of Hawaii. The system generates 30 random data points you see here in purple, which are then reviewed and marked with a pre-designated label. Prior to this internship, Malama Maunalua had no standard protocol for assessing these photos, so I developed one and selected 50 labels that could be used to describe what may be seen. Labels include things like different coral species, algae species, and substrates such as sand, coral rubble, framework, rock, and so on. Once all the photos are processed, the system can be used to easily locate sites that may be ideal for restoration work. The value of this study is that it contributes to a long-term monitoring project that Malama Maunalua has been conducting for several years now. Long-term data sets allow us to better predict trends over time so we can make the most informed management decisions. Malama Maunalua has been partnering with local nonprofits to rebuild reef structures in the bay as well. This information learned from the study can be used to select new sites for coral outplanting in which fragmented corals, or corals of opportunity, are established on these in situ beds where they can grow and help establish reef structure. Lastly, the fish data can be used as a baseline should a fisheries management area be established in Maunalua Bay, as was proposed in 2021. All of this said, it is really critical to continue monitoring efforts to get the best idea of where restoration and management is needed in the bay. This ecosystem has the potential to be productive again if properly managed. These are my references. And I'd like to give a huge mahalo to Malama Maunalua and everyone who assisted with these surveys. This was a really incredible internship opportunity, and I'm proud to have contributed to this conservation work. Thank you.